Assalamualaikum warahmatullahi wabarakatuh. I am Izzah Fatini binti Anuar, 1170839. In this video, I would like to present a very interesting topic, which is synthetic biology. This topic is known as one of the applications of biotechnology. Here, I will share three main components in this topic, which are the overview of synthetic biology by introducing the definition and the concept involved. Secondly, is the current news or research entitled Unilateral Gene Therapy Injection for treat to treat leber-hereditary optic neuropathy for patients who have vision loss about 6 to 12 months. And lastly, is the purpose or goals of this research. As an introduction, synthetic biology is a field of science that involves redesigning organisms for useful purposes by engineering them to have new abilities. In short, we can call it a nature-based design. Basically, in synthetic biology, it actually a study about how to build artificial biological system using a lot of same tool and experimental technique. The focus is often on taking part of the natural biological system, characterizing, simplifying them, and using them as component of an engineered biological system. Next is how synthetic biology occurs. It is by combining the chemical synthesis of DNA and the growing knowledge of genome. Therefore, it enabled the researchers to manufacture the catalog DNA sequence quickly and assemble them into new genome. Due to the increase of efficiency and cost of DNA production, allowing the scientists to construct and create engineered bacterial chromosome to be utilized for production of advanced biofuel, bio-based renewable chemical, bio-based specialty chemicals, for example, pharmaceutical intermediate, fine chemical, food ingredient, and in the healthcare sector as well. There are few examples of industry that using synthetic biology. For example, molecular biology, bioinformatics, computer science, physics, engineering science, chemistry, agriculture, and microbiology. For synthetic biologies, we can categorize them into two broad classes. First is synthetic biologies that use irregular molecules to imitate closely emergent behavior from normal biology to create an artificial life. Second, the one that seeking compatible component from the natural biology to combine them into systems that function abnormally. Okay, in December 2020, there was a current research entitled Bilateral Visual Improvement with Unilateral Gene Therapy Injection for Leber-hereditary optic neuropathy. So, this research actually was conducted by a team of scientists led by Patrick U. Wyman from University of Cambridge. Briefly, gene therapy is actually the insertion of gene into individual cells and tissues, while LHON or leber-hereditary optic neuropathy is known as the most mutual genetic mitochondrial disorder. Leber-hereditary optic neuropathy (LHON) is a maternally inherited blinding bilateral optic neuropathy. It is the most common primary mitochondrial DNA mtDNA disorder, affecting about 1 in a 30,000 to 1 in the 50,000 people, particularly young adult male. The pathophysiology of LHON is characterized by selective loss of retinal ganglion cell RGC and their axon, which lead to rapidly progressive bilateral vision loss. Recent treatment of LHON retained minimal IDB was granted market authorization in the European Union for treatment of LHON under exceptional circumstances. LHON is highly vulnerable to gene therapy because it has good onset with an expected natural history. Furthermore, the biological target, the RGC, are easily accessible for gene delivery through standard intravitreal injection. Besides, in this study, the use of unilateral gene therapy injection for 37 patients that lost their vision between 6 to 12 months. As I mentioned before, gene therapy is done by inserting the gene into individual cell and tissue to treat a disease and hereditary diseases by replacing a defective mutant allele with a functional one. In this study, the method of gene therapy is done by using a recombinant replication defective adeno-associated virus AAV, which is a promising strategy for monogenic blinding disease. The function is to transfer an altered complementary DNA encoding NADH ubiquinone oxidoreductase chain 4 NH4 into right eyes of the LHON patient. Besides, the specific mitochondrial targeting sequence MTS that is functional to allow the translocation of the mitochondrial matrix also contained in the injection material. The next part is the result of this study. At 96 weeks of the unilateral treatment injection was performed, the patient were analyzed and examined. Surprisingly, the treatment went very well, where about 78% or 29 patients portrayed a significant vision improvement in both of their eyes. Thus, the scientists did an investigation in non-human primate model known as Cynomolgus macaques because this species have a visual system that is close to human. Then they noticed that the viral vector DNA had moved from injected eyes to the anterior segment and retinal and optic nerve of contralateral non-injected eyes support a plausible mechanistic explanation for the unexpected bilateral improvement in visual function after unilateral injection. In a nutshell, gene therapy by a unilateral intravitreal injection with ND4 AAV have shown an improvement for visual function in LHON subject. Besides, the viral DNA transfer still need to be further investigation because it might have significant effect on the clinical trial design of the gene therapy and data measure. The team the team has set a goal and hope that this technique of maintaining the role of RGC and enhancing the patient side projection will be life changing, life changing for the patient, and the visual saving gene therapy become reality because the treatment, however, has been tested for safety, and the team is actively investigating the optimum therapeutic windows. Lastly, they also hope that this technique may be extended to the treatment of other categories of mitochondrial disorders. That's all for me from the explanation for synthetic biology. Thank you.